Hello, everybody, and welcome back to the second part of our live program in which our customers and collaborators are sharing their experience around our Oncomine solution for clinical research testing. So it is my pleasure now to introduce two more speakers. For this session, we will have Dr. Craig Mackinnon, who is Director at the Genomic Diagnostic and Bioinformatics Unit. He's also Professor at the Department of Pathology at the University of Alabama, Birmingham. He will be talking about utilizing the Genexus system to detect complex genetic variants in human tissue specimen. And right after this first talk, we will have another one from Dr. Jay Bayani. She's Principal Investigator Scientist at the Ontario Institute for Cancer Research. And she will be talking about the evaluation of the Oncomine Comprehensive Plus assay for profiling of solid tumors. So thank you both the speakers for participating in this event. Right after those presentations, we will have a Q&A session where the audience will be in the position of asking questions to the speakers. With no further delay, now I would like to leave the stage to Craig. Please go ahead with your presentation. Thank you, everybody, for joining.
Okay. Hi, everyone. My name is Craig Mackinnon. I'm from the University of Alabama at Birmingham. Here are some disclosures for today's talk. Okay, so today I'm going to talk about the Genexus system and go over the motivation that we at UAB had for transitioning over to the Genexus, and then talk about some research cases that we ran on the Genexus when we were evaluating it. And then I'm going to finish up with an interesting case describing a mutation in a human lung cancer specimen. So in a molecular oncology research lab, there's a couple of considerations you have to take into, you have to evaluate when you're setting up a panel. One is the target genes of interest for your panel. A lab needs to be able to detect a wide range of genetic variants. This includes single nucleotide variants, insertion deletions, fusions, and copy number variations. And as we all know, some of these variants are hard to detect. In addition, a platform that you're going to use in your lab also has to be capable of testing a wide range of specimens. Oftentimes, the specimens that are in your biobanks or that you're working with are very small. They can be very poor quality, oftentimes coming from FFPE, in which the DNA or RNA is degraded. The source can be a needle biopsy, a cytology specimen, cell-free DNA, and FFPE samples. So you want to find a platform that is really good at handling a wide range of specimens. At some, depending on what test you're using, a lot of times your sample isn't good enough and you might end up rejecting 5 to 20% of your samples. So given this, it's important to have an assay that can handle a lot of specimens. So you can think about really 3 different tiers of testing. Oftentimes you might have a specimen that's really tiny, and you might only be able to do either a single IHC test or maybe even just evaluate one gene, such as like KRAS or NRAS or EGFR. Or if your specimen is small but suitable for doing a panel, you might have to limit the number of genes you can have on your panel. And it can be a combination of DNA and RNA targets, allowing you to look for variants like insertion deletions or single nucleotide variants in the DNA, or fusions with using RNA. Or if you're lucky enough to have a large specimen, you can do a comprehensive panel that can include hundreds of genes and do the whole spectrum of mutations, including single nucleotide variants, insertion deletions, fusions, and copy number changes. At UAB, we had historically used a Haloplex target enrichment assay that had a fairly high requirement for our starting DNA input. The kit required 200 nanograms, but we had optimized it down to 100 nanograms. And the assay did have one advantage, is that each molecule that you sequenced had a unique molecular barcode, which was important for being able to have high accuracy and quality of your sequencing data. But one of the problems with this assay was that the starting material was so high that oftentimes we were unable to sequence a lot of our research samples. So that was one of the main reasons that started to motivate us to look around for another system. And we evaluated the Ion Torrent Genexus, and it had some features and qualities that were really important to us. One is it could work with a wide range of tissue samples. Most of our samples, research samples, are FFPE, and we have a lot of blood from those same samples that are paired with it. It works really well with low input DNA and RNA. Each pool that you work with only needs 10 nanograms. It can detect, depending on the kit or assay you're using, it can detect a wide range of variants, including S and Vs, indels, C and Vs, and fusions. Has very little hands-on time, which is really nice for the lab. And the two features that really were most important to us were the fast turnaround time of 2 days. And then we did some calculations and recognized that it represented a massive cost savings for us. This next slide goes over our cost savings. You see this table here, and this is an evaluation of running our current panel, which is a 100-gene DNA panel, compared to running the Oncomine Comprehensive Assay version 3. It's a research panel that includes 163 genes on the Genexus. And by doing a comparison of the two assays, we recognize that it would be close to $300,000 a year in cost savings, which basically would be close to paying for the instrument within 1 year. In addition to the huge cost savings, we also got a lot of new assays coming out of the Genexus. We could do RNA for this, which will replace some of our FISH assays, which are very expensive to run and less sensitive. And the faster turnaround time of 2 days was very, very compelling. And it also allowed us to capture a lot of the small specimens we weren't normally able to sequence. So during our evaluation of the Ion Torrent Genexus system, we looked at 2 of the assays that are available, the Oncomine myeloid assay, which targets 40 DNA targets and 29 RNA fusion genes, and the Oncomine precision assay. We found some of our old reference samples we had previously sequenced. We found 4 specimens we wanted to resequence that were DNA derived from blood, and 3 samples that were derived from FFPE that we were going to run on the Oncomine precision assay. So we'll talk about the myeloid Genexus assay first. So we identified samples that had 10 mutations in 10 genes that were very important in different myeloid disorders. And these genes could be categorized into different types of pathways in the cell that are important for cell growth. They're also very prevalent in different diseases that affect the blood, including myeloid leukemias and myeloproliferative diseases. And some of these genes represented variants that were really hard to detect too, either due to the size of the variant, such as in FLT3 or CALR, or ones that were just notoriously difficult to sequence because of the gene itself might have had a lot of GC-rich content, such as with CEBPA. So we thought by selecting these genes, it would really give us a good chance to evaluate the system and see how well it performs. So of the 4 samples we had, each sample had between 1 to 4 or 5 mutations in it. We ran each sample in duplicate. And so this table shows the 2 of the samples. The second column shows the mutation, the genes that we were targeting with the mutations listed, and some of the metrics from the sequencing run are described. And the next slide are our other 2 samples that had mutations in CALR, or a large set of genes. And again, the sequencing metrics shown here. And so this slide shows the results of running these same samples on a confirmation assay, which is the GeneStudio, which is a different platform. It runs the same genes, but in a different assay. And it's just to compare how well the Genexus assay does to the GeneStudio assay. So all the data here is summarized on this slide. And we can see for our 4 samples, we had an excellent concordance. Everything that we expected to be detected was detected on both the Genexus and the GeneStudio. A couple of points to make out here is on sample 2, we had two genes that had variants, the DNMT3A and the WT1. Our assay at UAB detected variants in these two genes are not covered by the Genexus or the GeneStudio assays. So we did not detect them, but we weren't expecting to detect them. And then one of our samples, sample 4, included a JAK2 mutation at a very low variant allele frequency, around 3%. And when we ran it in duplicate on the Genexus and on the GeneStudio, only one of the two samples were we able to detect the JAK2 mutation. And we attributed this to the very low variant allele frequency. So in summary, comparing the Genexus to our in-house assay, all the variants were detected as expected. And we had 100% concordance between the two platforms, the Genexus and the GeneStudio 5. So we're very happy by this performance. So now we'll talk about the Oncomine Precision Assay. We ran 3 samples through using the Oncomine Precision Assay to look at a range of different variants. The first sample we'll talk about is a case that had an amplification of the EGFR gene. Now, EGFR is overexpressed in a type of brain tumor called a glioblastoma, and that's an important signaling pathway that is believed to drive growth of the brain tumors. So we had a sample in which we had identified EGFR amplification. We ran it in duplicate, and this chart shows that on the Genexus, we were able to pick up EGFR amplification and recognize that in each of the samples, there are roughly 33 copy number amplification of the EGFR gene. Now, another interesting thing that came out of this experiment is we recognized that this same sample also had a loss of an allele of the CDKN2A gene, which we did not know because our assay at UAB did not target this. So in this sample, there was a gain of EGFR and loss of CDKN2A. Another variant we were interested in involves the MET gene. Now, the diagram on the right side shows in the upper part of the diagram what happens normally to the MET gene, and that's exons 13 and 14 and 15 are normally spliced together. And then the protein, after it's translated, gets inserted to the membrane, and the levels of the MET gene are tightly controlled because in exon 14, there's a recognition site for the CBL degradation system. And when MET levels reach a certain level, the CBL system comes along and degrades the MET gene product, and that's how the cell maintains the appropriate levels of the MET gene and can control signaling that way. Now, in certain tumors, including lung cancer, MET exon 14 is deleted, and that leads to exon 13 being spliced adjacent to exon 15. The consequence of this is that the CBL recognition site is now deleted, and that leads to accumulation of MET in the membrane and increased MET signaling, which promotes cell growth. So in order to detect this event, you can take 2 strategies. At UAB, we sequence the DNA. And when you do this, you have to examine both the splice acceptor and splice donor site of the MET exon 14, as well as the DRY sequence, which is the recognition sequence for the CBL degradation pathway. So there's a lot of real estate that you have to target, and sometimes you miss the mutation due to the sequencing issues. But we had a sample where we did find the splice site mutation, and in order to compare this, we would predict that due to the splice site mutation, the exon 14 would not be in the transcript. So from the same specimen, we isolated RNA, sequenced it on the Genexus, and identified that indeed MET exon 14 was deleted in this specimen. And that's shown in the lower table, where you can see in the column that says gene exons, MET exon 13 is fused directly to MET exon 15. So it's a way to confirm that both at the DNA level and the RNA level, the mutation leads to loss of MET exon 14. Now I'd like to finish this talk by describing a really interesting research case we had of a sample that had bilateral lung cancer in each lung, the right lung and the left lung. And when we sequenced both of these lung tumors from the same patient, we saw that on the right lung, there was a activating mutation in the EGFR gene. It changes the glycine at position 719 to an alanine. And in this specimen, the variant allele frequency for this mutation was 12.75%. We were really interested to also see that this specimen also had a T790M mutation at 6.2% variant allele frequency. And this is really unusual because T790M, you usually see that in the context of a lung cancer specimen that had been previously treated with drugs that target the EGFR pathway. So when we sequenced the contralateral lung, we found a different EGFR activating mutation, the L858R mutation at 6.8% allele frequency. And again, the same T790M mutation at around the same variant allele frequency. So this is really unusual, and we wanted to explore it further. And so something right off the bat, you're starting to think this is unusual, and it looks like this could be a germline T790M mutation. And in fact, when T790M is first identified in pre-treatment specimens, such as the ones I just described, about 50% of the time, this is a germline mutation. And these tumors often have a secondary EGFR activating mutation. And the degree of risk and penetrance for developing a clinical non-small cell lung cancer syndrome is unclear in these types of specimens. The behavior of these mutations is unknown. There's not many patients who've been described with that. Some reports say that this is an indolent clinical course, but that's not always the rule. And they often are associated with having multiple lungs tumors in both lungs, which is what we saw in this case, this sample. So this is summarizing a review article that looked at germline EGFR mutations, described 14 different mutations affecting EGFR, and 6 of them had the T790M mutation, as we saw in our samples. Now, it's interesting that in 5 of these 6 samples, there was a secondary EGFR mutation that was also described, similar to what we'd seen. So we really want to investigate this further to confirm that this was a germline T790M variant. So we resequenced the tumor DNA on the Genexus using the Oncomine precision assay. Okay. And so with the 2 specimens, one from the right lung and one from the left lung, we were able to isolate DNA only from the right lung. Unfortunately, the left lung specimen, it was depleted and there was no more opportunity to extract DNA. So if we're focusing just on the right lung, we expect to see the T790M mutation as well as the G719A mutation. Okay, so we sequenced on the Genexus, and this slide gives you an overview of the run level metrics that come off of the sequencer that allow you to assess how well the sequencing run worked. This diagram on the right side of the slide that shows a heat map of your lane, you can see it's red, which means that the lane loaded really well. The top left shows that your loading efficiency was 87%. There's about 1.5 gigabases of total sequence. Each sequencing reads about 100 nucleotides long, so you had 14,800,000 reads. And overall, the run looked really good. So this is a nice way to assess your run before you start doing your analysis. In addition to run level metrics, you can, for each sample, look at the sample level metrics. And that's summarized on this slide. And in this specimen, this one particular sample that we're looking at, there is 1.4 million mapped reads. The mean read length was 97 base pairs, and we had 98.4% uniformity. So these are very good metrics. Based on that, you have confidence that when you look at the readout from the variants that are detected in your specimen, you have high confidence that they're accurate. So this right lung specimen that had the T790M and the G719A mutations, we see here that they both that they have, instead of having 5% variant allele frequency, it's around 50%. So right now we know that there was something wrong with our assay when we ran it, that we were missing a lot of these mutant reads. And it was interesting to us that this patient also had a known polymorphism at the exact same variant allele frequency. That's the Q787Q polymorphism, which is just 9 nucleotides away from T790M. So we looked at the pileup of these two variants, and we can see that they're in cis with each other. So that means that every read at which we detect the T790M variant also had the polymorphic variant Q797Q, which is a silent mutation. It's not a mutation, it's a polymorphism due to a single nucleotide change. On the Genexus, the variant allele frequency for these two were identical, whereas in the UAB, the 790M variant allele frequency was only 6%. So clearly we missed a large fraction of the mutant alleles. So this shows us that the T790M was a germline variant. And we confirmed this by sequencing the blood that was paired with these specimens, and it also had a 50%, approximately 50% variant allele frequency. Reviewing the literature, there's a paper we discovered, published in 2007, that showed that when you sequence EGFR variants using cell-free DNA, germline variants have a variant allele frequency around 50%, which is shown by those green dots on the right bar graph, whereas somatic variants, which are depicted by the gray dots, have a much lower variant allele frequency. So the findings from this case is that all the expected variants were detected by the Oncomine precision assay, and it confirmed that the T790M variant that we identified was germline origin. The Q787 variant was not specifically covered by the OPA assay, the Oncomine precision assay, but these variants were covered by amplicons that are contained in the OPA. So by modifying the bed file, we were able to call these variants and confirm that both the Q787Q polymorphism and the T790M variant that we had initially detected both had a variant allele frequency of 50%. They were visualized on the same read, and it confirmed the germline origin of this variant. So in summary, we looked at 4 DNA samples derived from blood and 2 DNA samples derived from FFPE research specimens and 1 RNA that was isolated from FFPE. These were all run in duplicate on the Genexus. We had 100% concordance between the Genexus OPA, which is the Oncomine precision assay, and the Genexus myeloid assay with the UAB in-house Haloplex assay. Additional variants were identified. For example, we detected copy number variation of EGFR, and which was an amplification, and copy number variation of the CDK2A gene, which was a deletion. The sensitivity of Genexus assay was superior for some of our targeted regions, for example, the T790M, and we gained significant efficiency with turnaround time, hands-on time, and reagent costs. So that's all for this presentation, and if you have any questions, look forward to discussing them with you. Okay, so thank you very much.
Thanks very much for inviting me to share with you some of our experiences in the evaluation of the Oncomine Comprehensive Assay, plus for profiling of solid tumor samples. These are my disclosures. So the results of many translational research initiatives have been the identification of a number of prognostic and predictive biomarker. And this is really in response, as I said, to the translational research and drug development. So what is it that we need in terms of future precision oncology? And it's a delicate balance in obtaining assays that have rapid workflows, robust informatics and reporting, updated content and links to the literature and public databases. But we also have to be mindful of the fact that the samples that we're profiling are getting smaller and smaller. Some of the quality of these samples can be quite poor. We also need to think about the context of some of these tissues. So thinking about things like tumor to normal content. And once again, the uniformity in the informatic workflow. Not too long ago, we were in search of an NGS-based platform that could help harmonize some of the biomarker signatures that we developed, both through genomics and transcriptomics. And we came across the Oncomine family of profiling solutions. And we asked the questions whether the Oncomine platform could really satisfy our translational research needs. But before I go into the evaluation of this platform, I wanted to give you the context in which we were searching for this platform. So a lot of my work at the OICR focuses around improving the clinical management of women with breast cancer, specifically in the early hormone receptor positive breast cancer setting. So a little bit of the background: approximately 1 in 8 women, and much rarer in men, but about 1 in 800 men will develop breast cancer in their lifetime. Approximately 15% will die from their disease, and approximately 80% of the breast cancers that are seen in the clinic are hormone receptor positive. Unfortunately, despite adequate initial responses to that anti-endocrine therapy, approximately 50% will experience a recurrence. We had previously identified a 95-gene transcriptomic prognostic risk signature in a cohort of almost 3,000 patients. And what we found that there were a population of women who were at high risk for recurrence following an anti-endocrine therapy. The genes that comprise this signature were related to things like cell cycle, DNA damage, and many of these pathways are currently being targeted by therapies being used in the treatment of breast cancer. We know that transcriptomics is only one part of understanding the biology of these cancers, and we also have to integrate things like copy number as well as mutational changes. So in a separate study, we use a subset of that cohort to look at the copy number and mutational changes in genes and key signaling pathways, again, related to targeted therapies that are being used currently to treat breast cancer. And we found that the alterations in these pathways were also found to be prognostic. So to improve the clinical management of breast cancers in the early or even the neoadjuvant setting, we need more informative prognostic biomarkers for risk on anti-endocrine therapy. But we also need predictive biomarkers for the rational stratification to targeted therapies. And again, we want to be able to best match the therapy to the biology of the cancer. We believe that we have a comprehensive transcriptomic as well as genomic signature that can answer this clinical need. So what we need now is a platform where we can integrate these biomarkers into one platform and one workflow and help stratify these women not only for risk, but to specific targeted therapies. Another cancer that has a clear clinical need is pancreatic cancer. Although much rarer than breast cancer, 1 in 64 people will be diagnosed with pancreatic cancer. And as you can see by the estimated 5-year survival, the outcome for these patients is quite poor. The treatment includes surgery, chemotherapy, radiation therapy, and in some cases, in the context of clinical trial, there might be biomarker-directed therapies. The challenges for pancreatic cancer are slightly different than breast cancer. Although biopsy material is being profiled, these cancers have poor tumor cellularity. And unfortunately, there's the challenge of the poor outcome and survival times facing these patients. The OICR has invested in a large pancreatic cancer initiative called PanCureX. And the goal of this was to profile the genomic and transcriptomic landscape of pancreatic cancer. What I'm showing here are the results from publication about 2 years ago from the first 60-odd patients, where fresh frozen tissues from needle core biopsies were enriched through laser capture microdissection and extracted for whole genome and transcriptomic profiling. The median turnover from the time of biopsy through to the sequencing results was about 35 days, and it required anywhere from 50 to 100 nanograms of extracted DNA and RNA, as well as a matched normal for each patient. And I've shown here the average coverage for the tumor as well as the normal DNA. So to improve the clinical management of pancreatic cancers, we need to identify additional prognostic and predictive biomarkers. And the PanCureX and COMPASS trial initiative will help elucidate some of these key prognostic and predictive biomarkers. But what we also need now is a rapid and robust targeted assay and a requirement for minimal nucleic acid input. So this brings us to the Oncomine comprehensive assay and what our translational research needs are. And here I've shown some of the overview of content for the Oncomine Comprehensive Assay Plus and the workflow of about 3 to 5 days from the time of sample prep through to sequencing and through analysis. So what we're looking for in an assay is something that is robust and reproducible, has a streamlined workflow, rapid turnaround time, requires low input of nucleic acid, and is amenable to nucleic acids that are extracted from formalin-fixed paraffin-embedded tissues. We also need a workflow that has top quality informatics and reporting of the relevant variants, as well as up-to-date and clinically relevant content. So one of the first things we did when we looked at whether the Oncomine comprehensive assay or the Oncomine product line could satisfy our research needs, we did a reproducibility study. And for that, we extracted DNA from about 33 formalin-fixed paraffin-embedded, in this case, breast cancer tissues. The DNA was extracted, and that same aliquot of DNA was processed in 2 different laboratories. And as you can see here, these are the metrics from both sites, Site A and Site B, of the general QCs that come from running the assay. When we looked at the concordance between the variant detection and copy number detection, and these are based on the default Oncomine settings, we see that there was excellent concordance on those variant calls between the 2 sites and over 95% concordance on copy number calls. And as you can see here, in one site, the BRCA2 copy number loss was not detected, but when we looked at that data, you can see that the copy number was just outside the established threshold for the assay. So again, we were really happy with these initial results. We then wanted to look at the performance of the Oncomine comprehensive assay to other technologies. And this is because we're mindful of the fact that the data that we've generated across many of our projects have been derived from different platforms and different technologies. So when we wanted to look at the concordance between copy number, we had orthogonal data for some of these samples based on Affymetrix OncoScan as well as MLPA. And these represent some comparable bulk nucleic acid technologies. We also had orthogonal data in about 17 cases based on FISH. And when we look at the concordance, and again, excellent concordance between the Affymetrix OncoScan and MLPA data, and not unexpectedly, variable concordance based on the in situ or the per cell technology. When considering variant detection in a subset of samples, we had PIK3CA mutation information based on the mass spec-based assay sequenome, and again versus OCAV3. And when we use either one of the benchmarks, we found that we had 98% concordance for the detection of PIK3CA mutations across these samples. So not too long ago, probably about 10 months ago, Thermo Fisher released its newest kit on the block, which is the OCAplus assay. So once again, we wanted to look at the performance between its predecessor, OCAV3. And here we have the same samples that were run by OCAV3, and these are fresh frozen samples compared to those run against the OCAplus assay. And again, you can see here in our hands, we are achieving the minimal requirements for cutoffs for the different QCs for both the assays. And again, comparable results between the two assays. And when we look at the concordance for copy number, as well as actual mutations, and again, this is off of the default settings, we see that copy number and actionable mutations found in OCAV3 were also identified in OCAplus at similar variant allele frequencies or copy numbers. And what about FFPE DNA? These are the results from about 66 breast cancers that were profiled both on OCAV3 and OCAplus. Once again, we see comparable results in terms of the QC metrics for the sequencing. And again, in our hands, we are exceeding the minimum requirements for a successful run for these assays. And what do the results look like in terms of copy number and actionable mutations? Once again, we see excellent concordance between variants that were detected, again, overlapping genes between OCAV3 and OCAplus. And in fact, there is improved germline filtering in the OCAplus, and we were able to filter out some variants that were detected in OCAV3 that were passing the filter. And again, all copy number changes and mutations detected in both assays exist at the same similar copy number, as well as variant allele frequencies. One of the outputs for OCAV3 and OCAplus are whole genome plots. And what I'm showing you here, again, are the same samples, one processed by OCAV3 and one by the Plus assay. And you can see the extra content in the OCAplus gives a really good whole genome information, again, giving us information about the karyotypic complexity of these cancers. We also had Affymetric OncoScan whole genome SNP array data, and again, this is the same case. And we see that the Oncomine Plus profile gives a readout similar to what you would find using the whole genome array. My understanding is that in new release of the analysis workflow, OCAplus will be able to deliver additional information, including LOH. So we're looking forward to that feature in the coming months. And again, how does it compare to whole genome sequencing? So this is, again, the same case processed by whole genome sequencing and the copy number profile generated compared to OCAplus. And we see similar patterns of copy number gains and losses across this cancer genome. We then looked at the concordance in key driver genes, and this is based on the pancreatic, initial pancreatic data, again, whole genome sequencing versus OCAplus. And again, we're really happy to see that we're seeing over 95% concordance in terms of copy number, as well as concordance on the key driver variants relevant to pancreatic cancer. And I remind you that the depth of coverage for the whole genome sequencing is on average 50x. And again, just to summarize our OCAplus experience to date in archival tissues, we've now processed, since the release of this assay, 231 early hormone receptor positive breast cancer, and these are DNAs that have been extracted over 5 to 6 years ago from blocks that are originally processed over 10 years ago. From this sample set, 19 samples have been excluded from further analysis. And again, we followed the general QC metrics, and those cases that had greater than 2 QC metrics failing or falling below the manufacturer's suggested cutoffs were excluded. And we already excluded some of those samples due to poor library QCs. And again, this is suggesting that we just had poor quality DNA. In about 23 samples, they failed one of the, or fell below one of the QC metrics, suggesting that we might require a manual curation of the variants. But overall, we're really happy with our sequencing success rate of about 90%. And again, here are some of the QCs from these 231 samples. So the question is, where are we going with this? We're currently preparing a manuscript detailing a study where we performed OCAV3 in 387 early hormone receptor positive breast cancer. And the results here have shown that some of the copy number and mutational changes linked to signaling pathways are prognostic. The goal then is to validate the rest of the cohort, which is about 3,000 patients from this retrospective clinical trial using the OCAPlus platform. So we go back to our question, does the Oncomine platform satisfy our translational research needs? Is the assay robust and reproducible? Well, we've demonstrated that the same sample process at different sites using the manufacturer's suggested protocols yields the same results. Is the workflow streamlined, and does it offer rapid turnaround time? We've shown that the time to extraction, sequencing analysis could be achieved in as little as 3 to 5 days. We've also shown that you only need 10 nanograms of input per pool, so an average of 20 nanograms per assay. And this is in comparison to some other assays that require upwards of 50 nanograms. We've also demonstrated that the assay is amenable to nucleic acids extracted from formalin-fixed paraffin-embedded tissues. While I wasn't able to share with you some of the data from our other studies, we have been able to rescue samples that failed other sequencing technologies using the Oncomine platform and we're able to generate good data from those poor degraded samples. Is there top quality informatics and reporting of relevant variants? So we found that the variant calling for this assay is robust, and the software does provide flexibility to the user so that you can define your filters for a more customized approach to be able to answer the translational research questions for your study. One of the key things that was of interest to us is that variant calling is not dependent on a matched patient normal. The next factor is there up-to-date and clinically relevant content. So the current version of the Oncomine Comprehensive Assay Plus represents expanded gene content and signatures that are relevant to the current pan-cancer precision oncology research setting. And this has been determined by academic, clinical oncology, and pharmaceutical key opinion leaders. And we have been really lucky to have been working with Thermo Fisher and be able to provide input on the current content. Although I didn't talk about the Oncomine Knowledge Base Reporter in this session, this module is updated, used to be updated quarterly, but I understand now that it's updated monthly. So again, this is a wonderful resource that you can use in conjunction with the results that come out from this assay. And currently, is this technology available and widely used? Well, the ion Torrent instrumentation is widely used and is available for rapid uptake of new validated tests. In the last few minutes, I'd like to share with you how we're moving all of this translational work forward. About a year and a half ago, we were awarded a Genome Canada Genomic Applications Partnership Program grant. And this brings together an academic as well as a commercial partner to bring genomic solutions to market. What I've shared with you today are some of the clinical needs in some key areas, particularly in pancreatic cancer, breast cancer. And while I didn't talk about it today, we have identified a signature that can identify occult high-grade cancer in prostate biopsy samples. The deliverables are to bring an integrated NGS approach for each of these cancers and to be able to validate these signatures, as well as validating the platform to be easily disseminated to other translational research partners working in this space. So finally, I'd like to acknowledge all our collaborators and members of our team for the work that I've shown here today. Thank you very much.
All right. Thank you very much to both speakers once again for their excellent presentation. Appreciate them a lot. And we're now moving forward with the Q&A session. We have a couple of questions coming from the audience. We'll actually start with first with a question to Craig. Again, thank you for your presentation. It was extremely hands-on, so very useful, very practical. Appreciate it a lot. And the first question, it's about you built a business case about the number of hours and the time that you save when using the Genexus. Now, would you say that this is something unique, or that people could actually replicate that kind of business case and use it in their own setting, or is it because you have a special setting in your lab?
That's a good question. I think that the approach we took is easily translatable to any situation, because there's nothing really special or exceptional about the approaches we take. They're the ones that are used by most labs. I guess your probably your baseline cost is independent. It depends. Our assay is expensive. The reagents are expensive, and it's very time consuming because it's a kind of a first generation NGS assay. So your cost savings may be less if your existing assay is more efficient. But the Genexus is very standardized in terms of its cost.
All right. Thank you very much for your answer. I think our audience will appreciate it. There's a second question also for you, and this is about the T790M mutation, pretty typical resistance mutation of EGFR. And people are used to detect these when they do a follow-up liquid biopsy after, for example, have made the first analysis on the primary tumor eventually. So in your experience, so to say, how useful would it be the ability to use the same panel in tissue and liquid biopsy? Because that's what the precision assay is made for, basically to be used in both kind of tissue. Do you see this as a big advantage, or is just a nice add-on?
Yeah, I think it has an advantage. I think like with the newer drugs that are being used for EGFR testing coming out, the need to identify T790M is changing. So the more that you can detect from blood, the more spectrum of mutations you can detect, the better. And also too, and sometimes, and this is maybe talking, at translational research level, maybe even getting to the clinical level, which might be outside the scope of this talk. But some patients, you can't get another biopsy, that you're limited. So blood's the only thing you do have to work with. So by having a broader panel, that's very helpful. So I do think that the more you can do with a blood-based biopsy, the better for your research and the better for translational medicine.
Thank you very much again. Appreciate. Now we have a question for Jane, also for you. Thank you very much for your very informative presentation. First question is about the comparison that you have shown between the Oncomine Comprehensive V3 and the Plus. The question is about the input material that you have used. I guess the answer is yes, but this is the question coming in. You used exactly the same input for both assays, or the input was different?
No, we use exactly the same input. In fact, we use the same aliquot of DNA, and they would only be different in terms of time. Right. So there would have been maybe, for those who want to get really technical, one freeze thaw cycle, between the Oncomine V3 and the Plus assay. So they were exactly the same extraction, the same aliquot, just different times.
All right. Appreciate your answer. There's one more question for you. It's a follow-up question around this. It's again on the Oncomine Comprehensive Plus. In this case, you have shown the ability to detect copy number variant. The question is actually a little bit straightforward, so to say, or also a little bit, provocative in a way.
Yeah.
Is this really useful in the routine testing setting, having copy number variants?
Yeah, I think, I think it could help round out that, complete understanding of the whole cancer. It really depends on the context that you're looking, at this cancer. For us in the translational research space, sometimes you want to get an idea of what the rest of the genome is looking like relative to your, the variants that you're picking up, or the copy numbers that you're picking up. I go back, for me, it's really interesting to be able to see the genome because I go back to my roots of cytogenetics and all that kind of stuff. So I'd like to see what else is going on in the genome and not really hone in on the particular, the diagnostically important copy number changes, like 2 or MIC or something like that. I'd like to see the whole thing. So I feel like there is value in having the ability to see that whole genome.
All right, appreciate that. We have one more question for Craig, and this is actually about the ability to detect RNA fusion. How do you see, let's say, this evolving, and please, Jane, feel free to chime in as well. How do you see the evolution, let's say, between fish detection of fusion and NGS? Do you see a switch more into the direction of NGS, or do you see this fish is still, it has to be used as a orthogonal method for confirmation? Are we moving away from morphology-based testing in this case? I know pathologists won't be very happy to hear this. I'm never going to say it again. But again, how do you think this is gonna rule up in the future?
That's a great question, and especially we have a cytogeneticist here, so that'd be interesting. We've run FISH and have transitioned over to the NGS-based platform, and I think there's a couple of advantages. One, NGS can be more sensitive, especially on small samples. And sometimes you have samples, and you can use, for example, soft tissue tumors where you really don't know what the diagnosis is. So the FISH is being less of a confirmation study and more of a diagnostic tool to help you identify what the entity is. And you can do FISH NGS assays where you can detect obscure fusion partners too. So I think that it's nice to get away from FISH. It uses up a lot of tissue. It's very technically challenging. Depends on how good your lab is, how good your cytogeneticists are at making difficult calls. And it's a very powerful approach. So I think, if anything, it truly complements it and is a real workhorse in the lab.
All right, appreciate that. Jane, I'm not sure if you want to add anything on that.
No, I completely agree. I think there are definitely some advantages to the NGS approach, and there is value in FISH, but I think it is really more in the context of looking at the spatial relationship, looking at things that are really more in the research field, like the spatial heterogeneity of these fusions, for example. But if the point is to detect the fusion or detect these rearrangements, then NGS is really the way to go.
All right.
Yeah.
We have actually one last question, which is again for Jane, but Craig, I would appreciate also you if you wanna chime in. So the question is about if you had experience in sending out samples to provider, for example, send out, and what is the difference in the success rate between your in-house testing, which actually, Jane, you have shown to be really high, and your send out experience? Actually, I'm gonna have to say that we've been doing all of our sequencing, in-house. We've actually had some people send their samples to us to do validation, Right. So I think, and it goes both ways. We found variants that weren't detected through other sequencing modalities. And again, I think it speaks to the depth. This is a targeted panel, so you're getting, Right. Exquisite depth, in these targeted panels that you just don't get from, say, whole genome assays. So I think there's that sensitivity there. So we don't send a whole lot out, actually, anymore. That's okay.
Yeah,
perfect.
So, yeah.
Why not? Craig, I don't know if you had any experience in that.
Yeah, I think that usually, with most straightforward cases, there's a high level of agreement with send out and what we do in-house. Where you can run into some differences, I think, would be, for variants that are difficult to detect, like if the depth is much higher on one assay or another, that helps. But really another thing too is variants can sometimes be annotated differently on different pipelines from different labs, and that can sometimes cause confusion. It's the same variant, it's just described differently. And I've noticed that occasionally happens.
All right, appreciate your answer. So now I want to thank you both for your presentation, but also for taking the time to answer to this question. And I also want to close this session. Again, thank you very much for joining. Also, thanks to the audience for being with us. And it's not the end of the entire event. There's still something to go for, and it's the demo presentation live of the Genexus from our laboratories in California. My colleague Andrew is going to do that. And also, I would like to remind you that the entire environment stays open. So if you have the opportunity, please visit the hall of poster. There's a lot of nice, fresh data out there that might help you. It has been a pleasure to moderate this event. Thank you all very much again. See you next time.